Boy, I'm sorry I sound hoarse. I've heard Julia speak many times. I cry every time, along with many of you. I don't understand how families living with genetic disease go about doing what they're doing. Or Cameron, Julia, some of you in the audience. But the magic that keeps you going is the fire that keeps us working towards developing curative therapies. So this will be a boots-on-the-ground talk. I'm an academic, allegedly, but this will be a practical path forward to CRISPR Cures on demand. So I realized George Orwell is buried about 10 miles south of here, and I'm going to paraphrase him in speaking with you about the promise of CRISPR to create a nationwide solution to genetic disease. Genetic diseases affect all nations. But as I will argue to you today, while that's true and all nations are equal in that regard, some nations are a lot more ready to develop a nationwide approach to CRISPR Cures than others. And not to put a finer point on it, my number one choice for a country to do this in is the United Kingdom. And I will explain exactly why and what I think could be done and what we could contribute. So thank you, everybody, for putting this together, and it's such an honor to follow such august speakers. Here are my disclosures. So I have to start with the human component, as Lord Cameron said. This is a story of one child with a genetic disease that could be CRISPR'd but wasn't, and I'm convinced will be. It comes from Rebecca Arntz-Nicholas. She's a physician scientist at Children's Hospital, Pennsylvania. This child was born with a mutation in this gene, arginine synthetase 1, on the long arm of chromosome 9. And what you're about to see is the very many different mutations that cause this condition, and this child had one of them. So here's what happened to him. He was born and was immediately diagnosed with something wrong. He had high ammonia, which is horrifically toxic to his brain. He was transferred to CHOP. He was immediately diagnosed by whole genome sequencing as having this urea cycle disorder. And then I'm going to tell you the narrative of his life over the next 13 months. He was readmitted to the hospital. He came back to the hospital 6 times for recurrent episodes of hyperammonemia that poisoned his brain irreversibly. He had to have a gastrostomy tube. The standard of care is to do a liver transplant once the child weighs 8 or 9 kilograms, so you have to wait. So they did the liver transplant. It was associated with complications. But ultimately, here's where we are today. He no longer has a urea cycle disorder, but he's devastatingly developmentally disabled. He can speak in 2-word phrases and enjoys attending his special needs kindergarten. Today, if this child were born at Gosh, Greater Ormond Street Hospital, or CHOP, the standard of care would be the same. But what has changed between the story 6 years ago and today, and this is why it pains me as a gene editor to speak with you about this, and what we have to do in terms of, as Sir John said, moving scientific innovation into clinical healthcare, is gene editing. And the training wheels are off, and in fact, they first came off here in the United Kingdom. We can repair the mutation that caused this child such suffering with 100% certainty. I don't mean 99.7%, 100%. This is not hyperbole. This is my profession. We know everything how to do this. And this started with a clinical trial here, driven by an American company, Intellia, to repair a liver disease. And it's gone so well that they have now advanced to a phase 3 clinical trial. 760 human beings will be gene edited. This is how robust this is. Let me tell you specifically what happened, and then let me explain why Intellia nor any other company is actually working on diseases like that. I should also tell you that in the States, 2,500 children are born every year with conditions such as this one. So I mentioned to you that this story started here, and National Public Radio, which is, I guess, our version of the BBC, has a beautiful story about this, and this gentleman, Patrick Doherty, spoke about what it was like for him. So his father succumbed to this condition, TTR amyloidosis. It's a terrible prognosis, he said. It's just dreadful. Then he was a subject on the Intellia clinical trial for CRISPR gene editing, so single-time administration of CRISPR to knock out the toxic gene. And it worked magnificently. 4 patients were treated here, 2 in New Zealand. And Patrick Doherty was thrilled. He said, it's just phenomenal. Most importantly, and this is the real reason I'm speaking with you about this, is as NPR correctly pointed out, this is seen as a proof of concept that gene editing could be used to treat many other, much more common diseases. And my illustrious colleague, Jennifer Doudna, correctly pointed out that this is a major milestone for patients. So she's absolutely correct. But as I'm about to tell you, in order to make this sentence true, something needs to happen, and I will tell you exactly what needs to happen and why I think the UK is where this should be happening, and in fact, some of it already is. And to explain that, I need to tell you why Jennifer won the Nobel Prize. And she won the Nobel Prize for what you're about to see. So this is the heart of CRISPR. This is the protein Cas9. So you see a jumble of protein and nucleic acids. But the magic, even though it's scientific, is shown here. The way that this amazing machine recognizes its DNA target is using this RNA molecule to find a DNA that's complementary to it. And the rules of this complementarity were discovered by Watson and Crick based on data from Rosalind Franklin and Maurice Wilkins here in 1953. So this is a technology where you can go to a gene using rules that I can explain, I have explained to my 8-year-old daughter. And so the promise of CRISPR was shown in this Nobel Prize winning experiment where Martin Jinek in Jennifer's lab took a plasmid, cut it with a restriction enzyme, and then programmed the Cas9 to cut in 5 different positions of this plasmid using guide RNAs formatted according to these simple rules. And when he ran the gel, the enzyme lovingly obliged. I wouldn't be telling you this if this didn't go straight into Patrick Doherty. It's very rare that discoveries of this type go into humans unmodified. This one did, really. So this is Intellia's drawing of what went into this gentleman. The way this was mechanistically done is you take mRNA encoding Cas9, a guide RNA, it's 100 nucleotides, and you formulate it into a lipid nanoparticle. A lipid nanoparticle carrying mRNA. You will recall that Pfizer made 3 million doses of the SARS-CoV-2 vaccine using this technology. So this means that clinical delivery of CRISPR uses an approach that has been scaled to the planet. Now we just need to scale CRISPR Cures to the 400 million folks who live with genetic diseases, as Lord Cameron pointed out. So what happens once you inject it into Patrick Doherty or others? Well, the lipid nanoparticle homes to the liver, uncoats. The mRNA is translated into Cas9. Cas9 grabs the guide RNA, goes into the nucleus, and then uses the Doudna rules, the Doudna-Charpentier rules, to find its gene target, which in Patrick Doherty's case is a toxic protein, creates a double-strand break, and the gene is knocked out and the protein is gone. This worked better than, I think, even Jennifer Doudna's wildest dreams. And I'm going to show you just early-stage clinical data. This is the way the knockout would work. Cas9 will bind to the protein and knock it out. Gene, knock it out. But this is the real amazing part. So these on the y-axis is the levels of the toxic protein that killed Patrick Doherty's father, and this is the levels at the start. People get injected with CRISPR here, and as you can see, the levels of the toxic protein plummet to baseline at 1 month, and they stay flat. A single administration of a teaspoon of CRISPR, which vanishes in 3 days, creates a long-lasting elimination of the toxic protein from the bloodstream. This would not be exciting, except for what Intellia did with this next. And what they did with this next is they went after a different disease. Different disease, hereditary angioedema, different gene, KLKB1. How did they do that? By following the Doudna and Charpentier rules to keep exactly everything the same. They changed nothing except the 20 nucleotides that specified the gene target. So going to a different medicine for a different gene with this technology changes 20 nucleotides in 100 nucleotide RNA. And the clinical trial data are every bit as spectacular. 95% elimination of the protein which is being targeted out to 88 weeks post-dosing. What did I just tell you? I just told you that CRISPR is a therapeutics platform. In Jennifer's lab, Martin Jinek can reprogram Cas9 to cut naked DNA on demand. Intellia has done precisely this in humans. This is a family on the HAE trial. By using the Doudna rules to reprogram Cas, they've gone from treating disease 1 to treating disease 2. So why are we not seeing thousands of children being treated with this? I will tell you what needs to change. And I'm sorry, I don't mean to be Debbie Downer. I'm going to be Debbie Realist, because that will pave the path to what we should do. Intellia, unfortunately, while advancing phase 3s, had to cut research staff and cut its pipeline. The same is true for every other biotech trying to commercialize gene editing medicines. This is not just true for established companies. This is true for even newer companies. I will tell you exactly why this is happening right now and what needs to happen to fix it and where this needs to happen and how. Why is this happening? Because every time you change a guide RNA, it's a new medicine. And you have to go back to square one in terms of all the non-clinical effort. Let me be specific. This is Rebecca Arens-Nicholas. Let's say we make her a CRISPR that repairs the mutation in that child. We can do that in 2 months before his brain is damaged. And we treat that child with that thing, and the child experiences massive clinical benefit. A next child is born, but the mutation will be different. We have to swap out 20 nucleotides of the guide RNA. It's trivial for me to move this icon from one mutation to another, but that sets back the non-clinical path through regulatory and to the human, all the way back to square one, which for a liver disease means 3 to 4 years and industry prices $40 to $50 million. Who has the money to spend that much to develop de novo CRISPRs? And what about the 3 years? I told you, for this disease, 3 years is meaningless. All the damage is done in year 1. We have to get to this kid in the first 3 months. So I don't want to bring in an element of levity, but on some level I do, to showcase to you how ridiculous this is. I'm going to use an example of a biscuit I was introduced to yesterday by Stephen Saunders, a professor of pediatrics here in Oxford. They're delicious. They're called Jammy Dodgers, I'm told. And they are made, as I learned this morning, with strawberry jam, but sometimes people make them with raspberry jam. So imagine if you wanted to go from using one jam to the other, if you basically had to rebuild the bakery and recertify the flour, the sugar, the yeast, the oven, the packaging, the everything. And you're like, no. It's just the jam. Everything else is the same. Everything is safe. Everything's fine. We're just using different jam. But no. The rules are you go back to square one. This is what we're changing. So 3 years ago, in the New York Times, when I realized that this was happening, and I wasn't alone, the entire field was seeing this, I wrote an op-ed that we should be editing people, but we're not. And I argued that in order to build a world with CRISPR for all, fundamental change must happen. And at the time, the for-profit sector was already struggling. So I wrote a technical piece that I titled after my beloved John Lennon's song, Imagine CRISPR Cures, where I argued that the N of 1 genetic condition space cannot wait for the for-profit sector to work out its business model. I argued that we must engender change across the entire space in how, where, and with what financial support such treatments are engineered and delivered. I further argued that the academic nonprofit science sector can successfully innovate and develop administered curative medicine treatments. So all of these were words on websites. And for the first time, I'm very, not proud because nobody has been treated yet, but it's important for me to share with you that something has changed. And in our world, what has changed is a partnership between the Innovative Genomics Institute and Danaher. And what are we trying to do? We are trying to build a system that would address hundreds of genetic diseases using gene editing within a unified research, development, and regulatory framework. I'm going to run out of time, so I want to put the thank you slide now. So Jennifer Doudna recruited me to the IGI, and a remarkable team of physicians and physician scientists is working with me. Danaher has proven itself to be a marvelous partner and a company with a really good heart. And I just want to really honor 3 key people at Danaher Genomic Medicine: Sadiq Qasim, Vanessa Almendra Navarro, who's right here, and Chris Riley, among others. Without them, this would have never happened. I realized to my amazement that the partnership was called a beacon. If you take the Danaher flag and the IGI double helix and you superimpose them, you get a little beacon-like thing. So what are we doing? In order to explain to you what we're doing and why, and how this connects to, I think, what could happen here, I need to tell you about a cure for sickle and beta-thalassemia, and Baroness Blackwood referred to this earlier. So CRISPR Therapeutics partnered with Vertex to use the Doudna-Charpentier rules yet again to develop a medicine for sickle cell disease and beta-thalassemia. The way it is administered physically is a person comes to the clinic, their hematopoietic stem and progenitor cells are harvested, they're gene-edited ex vivo. The person going through the treatment, this is Victoria Gray, the first brave human to agree to do this, then gets chemotherapy and gets her stem cells back. And so just to inspire you deeply with what this technology can do and what we must do to make it broader in impact, let me show you the clinical trial data. So I'm going to show you what's called a swim plot. Every line is a human being. The vertical line is when they got CRISPR'd. And before these ugly objects are hospitalizations due to episodes of devastating pain. You need no education in medicine or science to realize how well this is working. Once you get CRISPR, they no longer go to the hospital. They get their life back. And the person who is the longest is Victoria, actually photographed here in England. And as Baroness Blackwood correctly pointed out, their extraordinary regulatory environment in the United Kingdom led to the fact that the first CRISPR medicine was approved here, followed by the rest of the world. So why aren't we celebrating? So let me tell you a story from Michelle Hermiston at UCSF, University of California, San Francisco. Although Ranjan Chakravorty from Oxford and Sophie Hambleton from Newcastle, who are both here, treat patients with this disease and can tell you directly what's happening in their practice. So I'm going to tell you the story of a child who died, even though he could have been edited to health exactly that Victoria Gray was. So the name of the disease is jaw-breakingly long, hemophagocytic lymphohistiocytosis. Let me just tell you what happens. So you start with a 26-month-old previously healthy child. Again, I don't want to start crying, but like if you think about the way Julia talks about what you witnessed happening to Emil, you have a healthy baby and you no longer have, and suddenly you no longer have a healthy baby. So they brought the kid to the hospital with fever and runny nose. It'll be still my heart, a 26-month-old has a runny nose. Then he starts to seize horribly. They diagnose him with this disease. It's genetic, it's a point mutation in a gene. They stabilize him, send him home, then bring him back, and he dies 44 days after diagnosis in the best ternary hospital in the world for this disease. So why did he die? It's the biscuits problem. Remember the biscuits, jam, strawberry versus raspberry? Every child is born with a new mutation. Every single one. Every child. To treat them, we need to make a new CRISPR. Who's going to invest in that? Well, as I'm about to argue, I know not who, but what country. So I want to paint a slightly broader picture. This is a relatively rare disease. There are 505 inborn errors of immunity caused by mutations in individual genes. The Model Foundation has a depressing table. It's 7 pages long, listing the diseases and the low numbers of patients with each one. But let's bring this back to the human element. This is Claire Booth. She's an amazing scientist, physician at Greater Ormond Street Hospital. And I asked her, Dr. Booth, how many patients with these do you have? And she said, April 2021, 1,285 with 559 with the more severe ones. So the technology to gene edit those people is fully developed, just like the technology is fully developed by Intellia to treat that kid with inborn errors of urea cycle. In fact, the number of these diseases, thanks to whole genome sequencing, is growing over time. Making the medicine using the Casgevy process is a solved technical solution. one biotech company is working on one of these diseases. What's going on? I will finally tell you why. It's actually a remarkably simple to understand problem, but it's not trivial to solve. And what I'm going to tell you is how we're doing this in collaboration with Danaher and why I think the United Kingdom is an absolutely unique place on earth to make this nation scale. So I need to tell you about work that the Innovative Genomics Institute led in partnership with the University of California, San Francisco, in developing a gene editing approach to treat sickle cell disease. The approach we took was to repair the causative mutation. Cas9, guide RNA, and then a repair template, a little strand of DNA. Break, repair the mutation, put the cells back in. Very proud to say the Food and Drug Administration gave us the go-ahead. How long did that take and how much did that cost? It took 4 years end to end, and academic pricing, $8 million. If you want to do this in industry today in the States, 40, because the industry prices are higher. Why am I telling you this? Because then I read this clinical story from Israel, and I read about a child in Raz Somek's practice who was diagnosed with a new inborn error of the immune cycle, point mutation, and despite the best care by a huge clinical team, died 10 months of age, new inborn error of immunity. And I read this and go, but oh my goodness, we can fix this using exactly the approach we developed for sickle. We can repair the mutation. So Dr. Somek calls Dr. Walters and says, hey, Mark, I have a kid with this. Can I fly you the stem cells? And what do? I just make the CRISPR. It's my job and that of thousands of others. We just make the guide RNA. There it is. That is the medicine that would have helped this kid. And we make this oligo. This process took me 5 minutes. Getting these reagents from IDT and Aldevron would have taken 1 week. But no. As I explained to you, if you change one thing, it's a new drug and you're back to square one. This is ridiculous, and this will stop. And the way this will stop is we will develop a platform for CRISPR Cures for inborn errors of immunity, where you do not go back to square one, where if you change one thing, the circle platform keeps running, leveraging everything you've done before. You do not rebuild the bakery, you just change the jam. The Center for Biologics Evaluation Research of the US Food and Drug Administration has recently put out guidance for how to build such platforms. I really appreciate the FDA for doing that, but basically what the guidance says, we don't know what a platform looks like, bring us some suggestions. We said, sure. The goal is this: if it takes you 4 years to get to medicine number 1, it should get you less than a year to get to medicine number 2, and less than 3 months to medicines number 3 through 48. We cannot do this alone. We're an academic university. We partnered with Danaher. We partnered with Danaher because the companies within the Danaher constellation, such as Integrated DNA Technologies, Aldevron, PNI Cytiva, Leica Microsystems, Beckman Coulter, and Molecular Devices, provide the entire constellation of reagents and technology solutions to build and de-risk a CRISPR cure. So we deeply appreciate their partnership. How are we doing that? You remember I argued for a key role of academics and nonprofits. We basically built an all-academic team of physicians, physician scientists, CRISPR specialists, and genomicists to wrap around this circle of a CRISPR cure with a focus on 2 inborn errors of immunity. One is this dreaded HLH, and the other is a severe combined immune deficiency. I would love to give you a full seminar about this, but instead, in the remaining 5 minutes, I will just say one thing and then explain in the last few slides why I think the UK is the place to scale this up to the nation. What's the limiting factor in saving a child in 3 months? Is that you rebuild everything from scratch. We have to have automated turnkey approaches where we just do step 7 or step 25 using a standardized approach we don't need to de-risk, that is run by dedicated personnel who spend their entire lives doing nothing but that. We have to rapidly, within a month, show that this CRISPR is safe. Right now, that takes a year. It needs to take a month. And so together with Danaher, we're building a platformized toxicity assessment that does not involve animals. Anytime you say the word animal, you're set back $300,000 in 6 months. The kid doesn't have that time. We will try to convince the Food and Drug Administration that if you change the guide RNA, you do not need to de-risk the oven. It's the same oven. But believe me, right now they will say you have to recertify the oven. But we'll say, why? It's the same oven. We've just changed the jam. The guide RNA is different. Everything is the same. Most importantly, and this is the heart of the story, right now, it takes $1 year and $1 million to make a clinical CRISPR, just the chemicals, the RNA, the guide RNA, the lipid nanoparticles. We have to get to a world where we can get to the kid before the kid suffers irreversible disability or dies. This is where the might of Danaher uniquely comes in. Let me give you some numbers. If you want to order from IDT or Aldevron CRISPR at research scale, it arrives for $100 in a week. If you want to comply with regulatory standards for what's called good manufacturing practice, and if you don't know what that is, you're a very fortunate human being. You basically add 3 zeros to everything, and you multiply everything by 12 months. It'll take you $1 year and $1 million. The kid doesn't have a year, and nobody has $1 million every time a kid is born with a new mutation. So in collaboration with IDT, we came up with, and this is a new term, phase-appropriate GMP manufacture that would take single-digit months and initially would be under 100K. What does that mean? I'll tell you what that means. If you speak with Rebecca Arntz-Nicholas or Michelle Hermiston or Claire Booth, what do they need the most? They need to be able to save the kid before the kid suffers irreversible disability or dies. If you have an open clinical trial for CRISPR in severe pediatric disease, and you have a newborn with a new mutation, phase-appropriate manufacture means you've made the CRISPR before the kid suffers irreversible damage or dies. By definition, and this is the central conceptual step, if you have safety testing of that CRISPR that has to take longer than the kid has, you have failed. Because the worst side effect is death from having not been able to catch the kid in time. And I'm humbled by the honor of the fact that IDT and Aldevron and Danaher have agreed to work with us on doing exactly this. In closing, surely you will say to me, this has been done before. Only twice, this CRISPR on demand to save a dying kid, and guess where that was done? Here. So this is Wasim Kasim. He's a physician who treats children dying of cancer. And you probably read the story. This is baby Layla being gene edited on demand before she dies of untreatable leukemia. I'm friends with Wasim, and he very graciously, I told him where I would speak and in front of whom. He wrote to the family and got permission to share this photograph. Again, I'm going to start to not cry. There she is today, just waving to us. Thank you for gene editing me. 6 years after she got edited. So this was done here. And do you know why this happened so fast? Because of the MHRA. The MHRA listened. And then the MHRA listened again. I spoke with Wasim about how he managed to save this kid more recently. Regulatory nimbleness and an understanding of what the benefit-risk considerations are. In conclusion, going back to inborn errors of immunity, whether the 559 currently in Claire Booth's practice or the manusage patients in Ranjan Chakravorty's or Sophie Hambleton's practice in Newcastle and Oxford respectively, we need to get to a world where as we diagnose these diseases using gene sequencing, the CRISPR cures arrive in time, so the cure space does not fall behind. So yes, platform processes, but as I just told you, the problem is that even though Casgevy is approved and editing is a platform, today the for-profit sector is struggling mightily because nobody understands how to take 3 years and $10 million to build a therapy for a kid who died 2 years ago. Nobody, but under the old system. I also said to you that innovative public-private partnerships, such as one we have with Danaher, create a unique opportunity to make an impact on diseases, for example, of the immune system, as I show you here, or inborn errors of metabolism. And so with that, in conclusion, if I were imagining a utopia where I would have the perfect country to make all of this real, what would I want? I would want the following. I would want a country which has a track record of clinical CRISPR. Ideally, it would be a country with a single-payer national healthcare system. And being American, trust me, I can explain to you why that is useful. We don't have one of those. It would be really great if that country had genome sequencing for diagnostic purposes deployed at scale and integrated with EHR. If only that existed. Now, the next thing I would need is a regulatory environment, so the regulator, the government regulator, that would be nimble to innovation in genetic medicine space. And finally, it would be really great if there was robust academic R&D innovation in genetic medicine space to basically leverage all of this. There's only one country in the world that fits those criteria, it's the United Kingdom. What to do? What is the actionable path to CRISPR cures on a UK-wide scale? I come with this with all humility, Prime Minister, leaders of entire organizations that are the size of the country here. So I say this with all humility but sincerity. Out of my window, I'm staying at Jesus College. I can see the Chapel of Exeter, and that's Tolkien. Why am I telling you this? Because after he studied at Exeter, he wrote The Lord of the Rings. And he introduced a remarkable concept, the Fellowship of the Ring. It's a constellation of superheroes with non-overlapping superpowers. This is why this works. Humans, wizards, elves, hobbits, and dwarfs bring non-overlapping superpowers. The fellowship of the CRISPR cure would need to be the same. We would need physicians who treat N of rare disease. We would need patient advocacy groups. We would need scientists, starting with people who sequence human DNA for a living and diagnose genetic diseases, people who really understand CRISPR, who can build disease models. We would need manufacturers of CRISPR components. We would need funding agencies of every flavor, from government to philanthropy to for-profit. We would need government regulators of experimental medicines, and then we need national healthcare system officials. When that fellowship of CRISPR cures is gathered, it can have a specific thing to do, which is, I told you right now, in the United Kingdom, there are thousands of human beings with completely treatable immune system diseases. One example, I do not want to speak about immune system diseases, the only one. There are a number of others which are treatable today. And a simple, actionable plan would be gather this fellowship, run on this platform process, and in 4 years, completely realistic number that, believe me, I've been doing gene editing for clinical purposes for 21 years. This is a defensible number. Treat 50 patients with 5 related rare diseases, each of whom has a different mutation. They're CRISPR treated in platform mode and critically from a commercialization perspective. This has to be a licensed medicine for CRISPR on demand. This can happen. I should also emphasize, and I think both Lord Cameron and Baroness Blackwood and Sir John referred to this in one way or another. You know, we talk about N of rare. Once an organ becomes editable clinically for a rare disease, we can rapidly progress to editing for more common diseases. I'll give you 3 specifics. In the lung, if you think about using editing to treat cystic fibrosis or primary ciliary dyskinesia, and I really want to honor and recognize Deborah Gill and Harriet Holm, who represent these 2 physician-scientist communities in the room for inspiring me to think about this. You can then transition that editing toolbox to pulmonary disease more broadly. If you treat inborn errors of metabolism, such as that kid who suffered in Pennsylvania, you can now treat metabolic disease. If you treat inborn errors of immunity, you can now treat autoimmune disease. But the unique power of this idea is once you have established the regulatory and manufacturing and reimbursement framework for this, you have de-risked all the bits and pieces, and everyone in the Fellowship of the Ring knows how to work with each other. I'm humbled by the fact that I got invited to speak before you. I will close with another quote from somebody who lived about 8 miles north of here. And I realize that it's a nuanced thing to quote him, but I've been quoting him for a long time. So Blenheim Palace is just 8 miles north of here. And Churchill said, I think during the war, he said, we can, we will, we must. And that, I think, is how I think about a UK nationwide project to build CRISPR Cures. Thank you.